Why do low protein diets work for weight loss? Some examples of low protein diets and approaches are the sugar diet, the Walter Kempner rice diet, which also has some sugars and juices in there, the potato diet, the Starch Solution, plant-based diets. Most plant-based diets are reduced in protein compared to omnivore diets. Fat fasts, juice fasts, and even all the different types of fasting. It is argued that a major mechanism of that is the restriction of protein. So people have started to focus on low protein ever since the popularity of the sugar diet, where people have been finding that they've been losing a lot of weight, even reversing poor metabolic health symptoms, having sort of quite startlingly good results that were unexpected for many. And this has led people to explore the whole idea of low protein diets. People on the saturated fat subreddit have been experimenting with low protein approaches and having some really impressive results. E.g. ex-fat loss, he's been doing a low protein, higher fat approach. What's Up Coconut, she's been doing a high carb, lower protein approach, and they're both getting good results. People who have been interested in low protein diets for reversing obesity and poor health have known for a while that it is to do with increasing FGF21, and this plays a key role in mediating those benefits. But what is less known is how the low protein diets come to increase FGF21 in the first place. There's a big part of this mechanism that I believe is missing. In this video, I'm going to present evidence for the theory that low protein diets increase FGF21 because of what they do to change the microbiome. Ultimately, it is the changes to the microbiome by low protein diets which are mediating the increase in FGF21 and all the health benefits. And this more clear understanding of the mechanisms involved can give us insights about how to truly optimize a low protein diet. I am going to mention the benefits of a low fat approach at the beginning, but then go on to discuss why often the low fat approach alone is not enough to truly optimize FGF21 and the weight loss and health benefits. In my previous videos, I have presented lots of evidence that obesity and poor health is caused by an overgrowth of problem bacteria in the gut. These problem bacteria primarily induce obesity and poor health because they damage the bile in the gut. This toxic bile then activates the bad bile receptor, FXR. Activation of the FXR bile receptor blocks the production of GLP-1 hormone in the gut, which ultimately induces leptin resistance, FGF21 resistance, and thereby obesity and poor health. Very low fat diets help to reverse obesity and poor health because they reduce the amount of bile that is present in the gut and available to get damaged by the bad bacteria. This is because it is dietary fat that is the main trigger for bile release into the intestines. Therefore, a very low-fat diet results in less bile being released and less bile being present in the gut and available to trigger leptin resistance, obesity, and poor health. However, as discussed in my previous video, it appears that the problem bacteria can also activate the bad bile FXR receptor independently of bile. For example, one study found that the bad bacterial metabolite agmatine can also activate FXR to induce obesity and leptin resistance. And another study found that hydrogen sulfide, another metabolite produced by the bad bacteria, can also reduce GLP-1 secretion by the gut and therefore leptin resistance and obesity. So it appears that the bad bacteria can induce leptin resistance, weight gain, and poor health symptoms all by themselves. They don't need to have this bile present to induce the formation of toxic bile, i.e., it may not be enough to just restrict dietary fat. A very low-fat diet may not be enough by itself. To truly optimize our GLP-1 secretion in the gut and therefore our health, we also need to clear the gut of the problem bacteria. It's not enough to just eliminate the bile. So, we can prevent bile accumulation in the gut with a low-fat diet. But how can we remove the bad bacteria as well to ensure that there is absolutely no blockade of GLP-1 production in the gut, and therefore no block to leptin sensitivity and weight loss? As I have explained in my previous videos, starving the problem bacteria of their essential nutrition is the best way to reduce their levels in the gut. Low carbohydrate, carnivore, and ketogenic diets do this by starving the bad bacteria of carbohydrates. However, if you are eating a higher carb diet, there is another way to starve the bad bacteria. The bad bacteria also require certain sulfur-based proteins to survive. So, if you are not starving out the bad bacteria with a very low carbohydrate diet, you can instead starve out the bad bacteria with a diet that is very low in their other essential nutrient, sulfur-based proteins. If you are starving out the problem bacteria with a very low protein diet, i.e. a diet low in these sulfur-based proteins, you don't also have to starve them out with a very low carbohydrate diet. Consider a plant. You can starve it of water and it will die. Or you can starve it of food and it will die. You don't have to starve it of both light and water. Either of those restrictions will kill the plant, and it's the same with the bad bacteria. You can either starve it of carbohydrates or you can starve it of sulfur-based proteins. Both will kill the bad bacteria. But it's not necessary to do both at the same time. You don't have to do a low carbohydrate diet and a plant-based diet at the same time. And this is why we get this observation that people on low carbohydrate diets can eat a lot more of those sulfur-based proteins, such as animal-based proteins, because they're already starving the bad bacteria of carbohydrates. They don't need to starve the bad bacteria of sulfur-based proteins. And then we get the other observation that people on higher carbohydrate diets do need to be a lot more strict with their sulfur-based proteins. This is why people on high carbohydrate diets get better results from more plant-based approaches, which naturally reduce the provision of sulfur-based proteins to the bad bacteria. Now let's look at the evidence. First, let me introduce you to a family of bacteria. They are the worst trouble-causing family of bacteria in the gut. This family of bacteria wreaks havoc on health in multiple ways, and they cause obesity. This trouble-causing family of bacteria is called Desulfovibrio. In 2023, the journal Microorganisms published a paper called Desulfovibrio in the Gut, the Enemy Within. Authors of this study stated that mounting evidence suggests a causal role for the overgrowth of Desulfovibrio bacteria in the development of various diseases. They presented a summary of research studies that have reported the effects of Desulfovibrio in various animal models in the context of the following diseases: metabolic syndrome and obesity, irritable bowel syndrome, irritable bowel disease, bacteremia, autism, Parkinson's disease, cancer, liver disease, kidney disease, autoimmune disease, and others. The researchers note that many studies have reported a positive correlation between Desulfovibrio and obesity and other metabolic syndrome phenotypes, and strategies which reverse symptoms of poor metabolic health also reliably decreased Desulfovibrio abundance at the same time. This could just be correlation though, so let's look for more evidence. Is the increase in Desulfovibrio just another symptom which correlates with obesity and poor health? Or are the increased levels of Desulfovibrio actually causing the obesity and poor health? There are several studies which in combination strongly indicate that Desulfovibrio bacteria are causing the obesity and associated poor health. not just correlating. Desulfovibrio are unique in that they are dependent on dietary sulfur amino acids, sulfur proteins, for their energy metabolism. A study published in the Journal of Clinical Nutrition in 2023 analyzed the association of obesity with the sulfur microbial diet in a large population-based cohort study. The sulfur microbial diet is a diet which is associated with 43 sulfur metabolizing bacteria, like Desulfovibrio, i.e., it is a diet which provides plenty of sulfur to promote the proliferation of sulfur metabolizing bacteria. The researchers found that adherence to the sulfur microbial diet was significantly associated with a higher risk of obesity and abdominal obesity. The researchers concluded that their results emphasized the significance of avoiding the sulfur microbial diet for obesity prevention, i.e., avoiding foods which are rich in sulfur amino acids. Desulfovibrio are a member of that class of bacteria which depends on sulfur amino acids for their growth and survival. The clue is in the name, Desulfovibrio. Therefore, restriction of sulfur-based proteins will starve this bad bacteria out. They cannot survive without sulfur-based proteins, so they fail to grow and survive, and their levels become greatly reduced in the gut. So, if Desulfovibrio bacteria are responsible for causing obesity and associated poor health, not just correlating, then sulfur amino acid restriction would starve out these sulfur-loving bad bacteria, and this would result in a reversal of obesity and associated poor health symptoms. And this is what we do find. Dietary sulfur amino acid restriction has been investigated in animal studies for 30 years and has repeatedly been demonstrated to have numerous beneficial effects on health and obesity. However, studies in humans have been limited until now. In 2024, a RCT study was published in the Journal of Translational Medicine, which tested the effect of dietary sulfur amino acid restriction in overweight and obese humans. Since animal foods are the most rich source of sulfur amino acids, the researchers used a plant-based diet as the sulfur amino acid restricted diet. And as we would predict, if these sulfur-loving bad bacteria are causing obesity and poor health, the researchers did find that sulfur amino acid restriction led to greater weight loss than compared to controls. They also found that sulfur amino acid restriction prevented the drop in metabolic rate which normally occurs as a survival response when people lose weight. And this is a huge part of why it is difficult for people to lose weight, because of this drop in metabolic rate. So the starvation of this sulfur-loving bacteria is preventing that from happening. It's stopping the body from fighting the fat loss, i.e., it is enabling fat loss. The researchers state that these findings are partly in line with animal studies where sulfur amino acid restriction increases 24-hour energy expenditure, increases the metabolic rate, despite losing body weight. They also noted a significant increase in fat oxidation, i.e. fat burning, after 4 weeks. And this fat oxidation remained elevated as further supported by the elevated plasma concentrations of ketone bodies in the sulfur amino acid restricted subjects compared to controls. These are all positive symptoms which indicate improved leptin sensitivity. So sulfur amino acid restriction in this study improved leptin sensitivity, presumably by starving out the Desulfovibrio in the gut. Another elegant study published in 2024 strongly implicates Desulfovibrio as being the root cause of poor metabolic health symptoms. This study didn't measure obesity, it measured another symptom, which is part of the same metabolic syndrome of strongly correlating symptoms, including obesity. A high-fat Western diet reliably causes obesity and liver inflammation in susceptible rodents and humans. This study measured liver inflammation with the aim of discovering the role of Desulfovibrio bacteria in the development of the liver inflammation. First, they fed mice a Western-style diet, and they observed that the mice experienced a notable shift in Desulfovibrio, an increase in gut bile acid concentrations, and increased liver inflammation. Next, to find out if the increase in Desulfovibrio actually played a causal role in the development of the liver inflammation and the changes to the gut bile profile, they then inoculated germ-free healthy mice who were on a low-fat chow diet. This low-fat chow diet normally keeps mice lean and healthy. However, the inoculation of the Desulfovibrio bacteria into the healthy mice on a low-fat chow diet was sufficient to cause the liver inflammation with no need for the Western diet at all. This shows that the Western diet only induces liver inflammation because of what it does to increase levels of Desulfovibrio bacteria. Because Desulfovibrio bacteria, even without the Western diet, is able to induce the liver inflammation. In this study, the poor metabolic symptom that was being measured, i.e. liver inflammation, was caused by the increase in Desulfovibrio bacteria as a result of the Western diet. The increase in Desulfovibrio bacteria was the cause. The paper also probed the question of how the Desulfovibrio caused the liver inflammation. And in keeping with the model of poor metabolic health that I have been describing in all my videos, the researchers found that Desulfovibrio caused liver inflammation because of what it did to change the bile in the gut. Desulfovibrio changed the ratio of different bile acids in the bile acid pool negatively. And as I have described in my other videos, this bad type of bile reduces the secretion of GLP-1 in the gut, which induces leptin resistance, obesity, and multiple symptoms of poor health. But a decrease in GLP-1 levels isn't the only way that high levels of Desulfovibrio bacteria and bad bile cause poor health and obesity. The damaged toxic bile it produces causes poor health in another way too. It's not just by reducing GLP-1 secretion. This toxic bile does not get reabsorbed well in the small intestine. Desulfovibrio changes the bile profile from one which is rich in taurochenodeoxycholic acid, which is readily absorbed in the small intestine, into taurocholic acid, which does not get absorbed easily. Therefore, this damaged toxic type of bile passes more readily into the colon, where it gets converted by more gut bacteria, in this very gut bacteria-rich environment of the colon, into the highly toxic secondary bile acids, deoxycholic acid and lithocholic acid. These highly toxic secondary bile acids are very damaging to the gut lining. High levels of toxic secondary bile acids have been found in the colon in colitis and colorectal cancer models. To make matters even worse, Desulfovibrio produces hydrogen sulfide, which also damages the gut lining. And, to add insult to injury, the Desulfovibrio bacteria also produce an inflammatory waste product called LPS. When LPS accumulates in the gut, it creates even more inflammation, which further damages the gut. So, high levels of Desulfovibrio bacteria produce high levels of toxic bile acids, an increase in bacterial metabolites such as hydrogen sulfide and LPS waste products, and this creates a perfect storm in the gut, which induces gut damage and leaky gut. This leaky gut allows the inflammatory LPS and the toxic bile acids to leak into the bloodstream, and this creates a storm of inflammation in the liver and throughout the body, causing even more negative health effects throughout the body. The researchers in this study also experimented with the sulfur amino acid restricting diet to starve out the Desulfovibrio bacteria. They also found that sulfur amino acid restriction was sufficient to reverse the liver injury and all the other pathological processes which were caused by both the high-fat Western diet and Desulfovibrio inoculation. Sulfur amino acid restriction lowered the gut bile acid levels, lowered the LPS levels, and enhanced the antioxidant and detoxification pathways. It is clear then that sulfur amino acid restriction does reduce levels of Desulfovibrio bacteria, and it is sufficient to reduce levels of Desulfovibrio bacteria enough to reverse liver inflammation and the bile dysregulation, which we know also causes GLP-1 deficiency, obesity, and poor health. So, if we want to truly optimize our GLP-1 production in the gut and prevent leaky gut and inflammation, we can do that in two ways. We can eat a very low-fat diet to reduce levels of bile in the gut, and we can starve the Desulfovibrio bacteria of sulfur-based proteins. So what would a diet look like that was very low in sulfur amino acids? It would certainly be a plant-based diet. Why? Because it is animal foods which are most rich in sulfur amino acids. All animal proteins, apart from collagen, are very rich in sulfur amino acids. So vegan or plant-based diets are automatically lower in sulfur amino acids. This fact explains why high-carb vegan diets and whole food plant-based diets have been found repeatedly to associate with better health and lower body weight in population studies when compared to high-carb omnivore diets. Although going plant-based is a step in the right direction, there are some vegan plant-based foods which are still also quite high in sulfur amino acids. Watch my next video to learn why the science is pointing to one particular sulfur amino acid, cysteine, as being the worst culprit, the important sulfur amino acid that we need to restrict to starve out the bad Desulfovibrio bacteria. Thanks for listening, and hopefully see you in the next one.